This is my presentation, Taming the Wild Morel. I'm Michael Scott Zerpoli. Before I get too deep into my presentation, just a little more background information about myself. I grew up in Northeast Pennsylvania, and I moved to Washington in 2011 to pursue a degree at the Evergreen State College. After taking a couple years off in working in the field of early childhood education, I came back to Evergreen in the fall of 2016 when I took the Fungal Kingdom with Paul Przybylowicz and Lolita Calabria. The following spring, I was the teacher's assistant for spring fungi, a class I was also enrolled in. And during that class, I worked with Noel Macnicki, and I was really lucky to get to go down to Trout Lake and do some morel hunting with Mike Bugh, who is arguably, in my mind, the unsung hero of morels. He's quite the expert. I'm very lucky to have had the opportunity to work with him and meet him. He has really been the foundation for everything I've learned about this mushroom. During that same quarter, I started working with Lolita on some advanced undergraduate research with my research partner who's tuning in tonight, Heidi Steinbach. We were investigating the impacts of aqueous reindeer lichen extracts on prairie plant seed germination in the Mima Mounds here in Olympia. That research continued through the next year with funding through the Stunts Foundation, and at the same time I worked with Paul Przybylowicz, building a culture collection for the Evergreen State College to act as a resource for future mycology work at the school. The following fall, I came back and TA'd for Fungal Kingdom again, and then Paul and I kind of got together and we worked with a group of students through the following winter and spring on like the initial stages of this research into morel cultivation that I will get into a little bit later in my presentation. So it's been a long journey at Evergreen. I've spent a little more time maybe than I should have there, but there's just been so many opportunities to work with really great people and to progress my experience not only in like the lab sciences and in research, but also in teaching. I could give a 45-minute presentation on Evergreen. I am going to save you from that, but I am always happy to talk about that place. So morel science, essentially, up until very recently, as much, I think anyone that's familiar with morels just through hunting knows that there's a lot of mystery surrounding them. There's a lot of like suggestions that some might be mycorrhizal, some are saprophytic, so they're like primary decay fungi as opposed to like associating with trees. It's just been a bit of a mess, and this has been in large part due to kind of an issue that's existed in mycology as a whole, which is the whole splitting versus lumping. And anyone that's not familiar with taxonomy, this is just basically the idea of, to what degree do you let details dictate speciation amongst a fungi as opposed to kind of just looking at them. I have this word here, ubiquitous, as an ubiquitous organism. And for a long time, morels kind of were viewed through that lens. A lot of the scientific publications kind of regarded only 3 morels based mainly off of their color. We now know, due to some pretty intensive taxonomic research, that there are at least 60 morels worldwide. And there's a lot of overlap in their distribution and in their life cycles and in their ecologies, but at the same time, there's a lot of differences. And as you'll see later in the presentation, not having a clear idea of morels on a species level actually really acted as a hindrance to the progression of the science. Since we have kind of resolved this taxonomy, and in addition to this cultivation boom that I'll talk about that's been going on in China, we've been able to answer questions about morels just in the past 5 years that we could never even get close to answering in the previous 100. And here, Lauren shared this during the last meeting. This is just kind of a breakdown of the morels that are confirmed to exist in the Pacific Northwest. I say that because there are probably other species that are here that don't have type specimens, that aren't well described enough to have been given a species delineation. This list, I know the first time I saw it, I didn't care too much. It seemed insignificant to me. But the more time I've spent with this list, the less mysterious morels have seemed to me. And I think that's because as you learn particular species, you realize that they fruit at slightly different times or in slightly different areas. So whereas, 20 years ago we thought it was one morel doing all this different stuff, we now know it's a cast of species appearing at different points in time. It can really change the mystery around the mushroom and help us like focus our intention on them and kind of make us less restricted in our ability to hunt these mushrooms. I think old wives' tales kind of like dictate our morel hunting patterns a little bit too much. And when you actually use this framework to investigate the mushrooms, they're slightly less mysterious. And I will say, even though it took some intensive DNA analysis to get to this list, only 2 species, Exemia and Sextillata, actually necessitate DNA analysis to differentiate. So you can actually just tell all of these morels apart except for those 2 just based off of where you collected it or some morphological features. So it is something worth investing your time in if you have the time and energy and you're interested. It's a daunting task, but it can be done. Before we get into talking about morel cultivation, I just kind of wanted to throw in this picture of some proposed morel life cycles that have come up at different points in time. Both of these tried to act as like a unifying life cycle, essentially trying to describe like all the different things we were seeing in different species of morels. As we now know, since there's a lot of different morels, this is pretty theoretical and really shouldn't be taken with more than a grain of salt, but I think it does highlight the complexity of their life cycle compared to other mushrooms that we might be more familiar with growing. These 2 terms are going to come up a lot when you're reading about cultivation of morels. I'm going to use them throughout my presentation, so I just wanted to make sure you are familiar with them. The first is Constantinella, which is the asexual stage of Morchella. This was originally described as an independent fungi. It presents as a powdery mildew. You probably wouldn't think much of it when you walked by it in the forest. You probably have walked by it in the forest. And it wasn't until we looked at DNA sequences that had been taken from Constantinella samples and compared them to Morchella samples that we realized they were actually the same fungi, which happened with not just Morchella, but a lot of mushrooms when we started using DNA to analyze them. So this is an asexual stage of morels. We don't really know the role it plays in the natural setting. Even though we know they're the same fungi, it wasn't until this past decade that a scientist at Washington State University actually sequenced Constantinella from the natural setting and was able to compare it to fruit bodies occurring the next spring and confirming that they were indeed the same thing. And that is the only instance in the scientific literature of someone linking the asexual stage to the sexual stage of Morchella. We mostly know about Constantinella from its appearance in burns and in cultivated sites, especially in China. And there's been a lot of research in China into the role of Constantinella. A lot of that work, unfortunately, has not been published at this point in time in English. So I just wanted to kind of give you a visualization of Constantinella. Excuse me. These were points in my yard that I inoculated with some Morchella spawn earlier this winter in January, and within a few weeks of putting it into the soil, I was just trying to assess the viability of the morel spawn in non-sterile soil. Within a few weeks, you started to get these white growths here on the surface of the soil. It spread. It continued to spread, I'd say, until about the end of April, and I found it up to about a half meter away from the points of inoculation. And I found it in a lot of situations associating growing on, I'm not entirely sure. I wasn't able to do the microscopy because of the whole situation with the virus, but it wasn't uncommon to see the Constantinella growing on plants. And you'll see that again in this picture, it's covering a leaf. This was actually a spot where I had harvested morels the previous spring, and returning in the fall, I found this Constantinella, and using microscopy, I was able to confirm that it had the same structure here that Lori Karras had described in her paper in 2017. I have samples for DNA analysis at some point in time, but I also found it occurring on the organic farm this year in association with sexual fruit bodies, occurring at the same time as the sexual fruit bodies. So I was picking morels, and I kind of dug into the wood chips a little bit and pulled up some cardboard, and this Constantinella was all over it. Again, confirmed with microscopy and all that. So my reason for really showing this to you, aside from its importance in cultivation, if you know how to look for this stuff, it can really inform your hunt. I've gone out to burn sites weeks before I found mushrooms there and observed Constantinella and knew to come back to that spot in the future. And at this point in time, it's been a consistent way to find morel spots out of the season. And I should mention, when Lori found hers, it was in the fall after a fruiting and before a fruiting, so it typically doesn't only occur at the time you're finding the mushrooms. Sclerotia is a very important factor in morel cultivation, and it's actually theorized to be the most important factor in cultivation. They act as these nutrient storage, not only for morels, but other fungi form sclerotia. They're typically formed in the soil. It's not uncommon, especially in burns, to find them kind of encrusting the top of the soil. They form pretty discreetly and then coalesce into these larger masses. These are malt extract media petri dishes that I had grown sclerotia out on for some of my work. And it wasn't uncommon to see them at points in time producing metabolites. And the more I dug into the research, it became apparent that sclerotia actually goes through several maturation phases, and it's not uncommon for it to just form more mycelium that goes into an asexual stage and then to continue to develop. I mean, this stuff will grow out, bring more nutrients back several months after it initially developed. So it is like a long-term, it's been theorized to be an overwintering structure. It's been theorized that this is kind of what takes seat and burns immediately after the burns, and then it overwinters and leads to the production of the mushrooms. These are things that we really can't answer. These ecological questions, like the role Constantinella plays in the life cycle of morels, or the role of sclerotia, these things are incredibly hard to study in the field. I mean, anyone that's been out hunting morels know they do have a pretty limited season. It's hard to do field work. It's hard to get into burns and do field work. And a lot of that has limited the progression of our understanding of these mushrooms. And by being able to cultivate the mushrooms in the lab, it gives us an opportunity to understand them on a level that really couldn't be done in the natural setting. And this is really kind of just, I don't want to say anything. I know there's avid morel hunters. I am one myself, and this is something I've kind of had to sit with for a while now and think on. But as I came to the realization that there's more morel species with limited distributions than we ever thought, it made me realize that we truly don't understand the roles that these mushrooms are playing in ecosystems, and we really don't appreciate how rare some of these mushrooms may in fact be. So I think when we go into reestablishing environments, not only are we, putting everything else at risk, all the plants, everything else that's trying to make a comeback, we are definitely depriving that ecosystem of something that we don't understand enough. I can tell you from experience, Other things are eating morels out in the woods, animals, squirrels, bugs. Morels can persist for a long time in the environment. So when we go into a burn area and pull out every morel we can carry, we might be doing more damage than we realize. Cultivation also gives us the opportunity to reduce the market price of morels. I think morels, as delicious as they are, anyone that has found them in abundance may question why they sell for so much. They might have a limited season, they might be hard to get, but it's something that I think everyone should be able to enjoy, and kind of the allure we've built around them has driven that market price, and I would like to see that go down so everyone can enjoy the mushroom. And it's been particularly challenging to cultivate morels, one, because they are ascomycetes. Most cultivated fungi are basidiomycetes, your cap and gill mushrooms, whereas these are your cup fungi. Just a handful of other ascomycetes have been successfully cultivated, and the protocol for those is just so different from what we use to grow other mushrooms. It's also made it difficult because, like I've talked about, the poor understanding of the life cycle as well as the taxonomic confusion. That will become a little more clear here with the indoor cultivation stuff, but that was one of the biggest roadblocks in progressing this field. In addition to that, spoiler alert, but morels have been successfully cultivated, but the protocol that details the processes for cultivation have been issued as patents. There are no instances where it has been presented as scientific work or free information. So I can say at the best, it puts you in a situation where there's a lot of guessing. And that's, you'll see what I did with my research. I just had to spend a lot of time trying to fill in blanks in the protocol. And this is kind of fueled, especially for the indoor cultivation, by the story of Ronald Ower, who is the first person to ever successfully cultivate morels indoors. And Ron was working as a university student in San Francisco in the early 80s, collecting morels from natural samples and culturing them, comparing biological characteristics, and attempting to cultivate pretty much everything he collected. And after years of work, he succeeded. He, due to financial constraints, decided to pursue patents for the work he had done as opposed to publishing detailed protocol. And unfortunately, 2 weeks before the patents were issued, Ronald Ower was mugged and murdered in a park in San Francisco. So essentially, we ended up with a situation where the only person who knew how to put Ower's protocol into practice were some of the venture capitalists he had been working with, namely Gary Mills, who took control of the patent and moved the industry to Michigan. And this is where the story, I mean, maybe when we get to questions, maybe someone that's in the Midwest or has a little more familiarity with the mushroom industry in the 90s, I see a lot of reports of a successful industry in Michigan in the 90s and the early 2000s. There was claims of wanting to ship 3,000 pounds week to week, coast to coast. But I can tell you there is not an active industry in Michigan now. Mills still controls this patent. And in addition to that, he's recently published work kind of analyzing microbial communities of substrate during fruiting, which tells me that he can successfully apply these methods to cultivate morels. Two scientists who worked very hard on replicating Ower's work were Volk and Leonard, who worked in the Midwest, I think at the University of La Crosse in Wisconsin. These two fell into a bit of a taxonomic trap because what had happened was Ronald Ower had identified the morel that he cultivated and issued these patents under the name Morchella esculente, which is the blonde morel. It's the morel that's, hypothesized to associate with hardwood trees. It's now called Americana. And being in the Midwest, I mean, anyone that is out there knows Americana yellow morels are more abundant than we can find here in the Pacific Northwest. So Volk and Leonard were in the perfect place in their minds to do this work. And after several years of attempting with Esculente from cultures they had collected themselves, they were not successful. in producing even primordia with Escalente, and they moved on to other morels they had collected from natural settings, including black morels, and they saw more success with some of these other species. They were able to at least get primordia to form. And what Volk and Leonard really did was kind of fill in some of that like fundamental uh, science that was missing from Ower, because again, the venture capitalists took control of the patent and not much work was done to explain the science behind what was happening. Uh, in 2005 to 2010, a scientist in Israel, Sigal Masfany, adapted Ower's protocol to cultivate Morchella rufibrunea, which was a morel that was new to science as of 1999. It was described by Guzman. It has a very limited distribution naturally. It has been spread more worldwide since then due to human intervention. But naturally, it occurs southern California, down into Mexico and Hawaii and in these other more temperate places. And what happened was Ower had actually collected and cultivated rufibrunea. And this was confirmed in 2011 when all of this taxonomy revision was going on. So that, created a situation where Volk and Leonard were chasing their own tail trying to cultivate the mushroom that Ower had identified, not realizing that, if Ower had been 50 miles north up in the Gold Coast area or even up in Oregon, he may have never run into rufibrunea. At that point in time, it wasn't really occurring that far up on the West Coast. And this is the basal morel. This morel actually differentiated out over time after evolving during the Jurassic period to form all other morels that we have. And there's a lot of overlap in its ecology because of that, as well as its morphology. So it's not surprising that he misidentified this mushroom because we didn't even know about rufibrunea then. But it created this situation where some folks spent almost 20 years trying to cultivate the wrong morel. And now that we have resolved the taxonomy a bit more, it becomes obvious that Escalente might not have been so good because it's probably mycorrhizal, whereas rufibrunea is a saprophytic species, which all of our cultivated fungi are. And then the only other reported success of indoor cultivation of morels was an adaptation of Chinese methods for outdoor cultivation that was done at Penn State by a master's student in 2019. She was also able to get primordia to form for Morchella importuna, which is the landscape morel that we have here most predominantly in the Pacific Northwest. It's the morel I focused on in my research. She was not able to get those primordia to mature. And that is something that Volk and Leonard spent a lot of time covering, was that it's really, really easy to get sclerotia to form more mycelium and go through asexual stages. The conditions needed to get it to produce sexual fruit bodies, even primordia and then let alone mature, is just, we truly don't even know what those conditions are at this point in time. But a brief overview of Ower's method. On a theoretical level, Ower came to the conclusion that morels produce their fruits in a nutrient deficient layer. So essentially their mycelium will grow out into the nutrient-rich spaces and translocate those nutrients back to sclerotia in nutrient-deficient areas, and then after it's been able to tap into everything it can get to, it produces mushrooms so it can, let off spores and get to another nutrient-deficient, nutrient-rich interface. I think this is why you see morels a lot of the time on trail edges in the forest, because those trails act as somewhat of a nutrient sink in the forest, right? And based on this method, based on this theory, Ower came up with a method to produce sclerotia in mass, which he determined was necessary for successfully cultivating these mushrooms. And he did that by taking some soil, a layer of tinfoil, and then just some grain. And he did it in jars because this is the 80s, and the most popular methods for mushroom cultivation. He was trying to adapt psilocybe cultivation to morel cultivation, so he started working with jars and ended up with these two layers, which I have a picture of here in a second, And what happens is the sclerotia forms in the soil on top of the jar, and then you take all that soil and sclerotia out, and then you put it into more nutrient deficient substrate. And this is where, like, one of those points in the protocol that is just so vague, in the sense that Ower's like, you can use any kind of potting soil essentially. I don't know how true that is. We obviously need to do more work to figure that out. I do know morels prefer to grow in slightly acidic and sandy soils. And then after this period, there's two variations in Ower's protocol where you can either grow more sclerotia initially and then plant more, or grow less and feed it more nutrients. And the only reason I'm bringing this up is because you'll see what happens when you add nutrients when I get into my own research. And after this period of inoculation, you allow for brief colonization, about a week, according to his protocol. And unlike basidiomycetes, your substrate doesn't become completely bound by the mycelium or anything. In fact, it's still pretty loose, but you will see the mycelium kind of wisping through everything. And then after it fully colonizes, there's this period of heavy watering, which again, the science behind why we water the sclerotia, why we water our trays at this point in time, isn't really well explained. But according to Ower, a week after this heavy watering, which occurs over a 24-hour period, you see primordia formation. And then all you got to do is find that perfect sweet spot that the morels will actually fruit in. And that's something that Ower didn't stress as being that important, but at the same time, it really seems to be everything. So here are some of the jars I was talking about. That's the soil on top, the grain on the bottom. This was the start of sclerotia formation. This was after a month of maturation in the jars. So all of this was just collected and used as inoculum. Now, the outdoor cultivation history pretty much like most of the outdoor cultivation work has happened in China. There were some early reports of success cultivating morels outside in Europe around the turn of the 20th century. But in all actuality, there's so much contention around those reports, whether they were just naturally occurring morels or actually cultivated morels. So it's just something I put in here because it's worth noting that people have been working on this since 1882. But it wasn't really until 1983, in the 80s, that some Chinese scientists started to report success with field methods for cultivation. Again, you have people reporting success, no detailed protocol. One of the scientists reported successful primordia cultivation or primordia formation, but didn't see the primordia mature into full-grown morels. It wasn't until the 90s that a scientist actually kind of cracked. I mean, it was more of an accident than anything. The story that I've read is after years of attempting outdoor morel cultivation, someone left some nutrient bags that they were using, kind of trying to replicate Ower's idea of feeding additional nutrients to build up the sclerotia, and they realized that morels fruited around the bags. And they developed these methods based around this idea of placing these exogenous or extra nutrients out into the field after inoculation, kind of continue feeding the mycelium and leading to fruiting. And these methods work well enough that there is an entire industry at this point in time in China based around cultivating morels in this fashion. There are some issues with this, though, especially in terms of sustainability. Again, the application of the nutrient bags is considered the most important breakthrough in outdoor morel cultivation. And it is based on that idea that you add the bags, feed the mycelium, remove the bags, create a comparatively nutrient deficient substrate. That's what the methods say. And I'm stressing this because this picture here is of a field and farm in China where you clearly see the morels fruiting in the presence of the nutrient bags. And this is something that I saw, this idea of nutrient deficiency, nutrient rich, and how sclerotia only form in one place. I found it not uncommon to see sclerotia forming a nutrient-rich substrate. It's obvious that morels can fruit in nutrient-rich substrates if this soil is being fed extra nutrients and you're seeing mushrooms fruit there. So again, we have something that works, but the science is not very clear as to why it works. And because of this, there are some real issues with sustainability. I mean, you'll see reports of yields of up to 8.4 tons per hectare. And then in those same reports, you'll see that some farms are getting 0 tons per hectare. And you're finding situations where farmers that use fields multiple years in a row essentially just fry those fields and nothing will grow there. It's one of those things that seems to be moving in farms set up for this. There isn't a lot of great success, and farmers are struggling to kind of make this a real livelihood. So, and a lot of that is based on the fact that even though this is like artificial cultivation, it is bionic in the sense that they plant these morels in areas that you would hope to find morels anyway. A lot of it's up in the mountains above a certain elevation. A lot of the time they're frying the fields beforehand, essentially kind of like with chemicals to replicate disturbances in the microbial community. They found that works, but then it reduces yields in the future, just like is the case with burn morels. So there's issues with this whole setup, and in a lot of cases, people are not being able to actually do this as a livelihood. But they've done it, and it's impressive. It's impressive to see, and it's something that we're only going to get a clear idea of how it works by attempting to bring this process over here in the United States where we can study it more closely, and that's a lot of what my research was focused on. There is just a list of successfully cultivated species at this point in time. You can see our importuna, our landscape morel, and rufa bruni is there again, and then burn morels have been shown to grow well in these conditions. So that's kind of where they're at with morel cultivation at this point in time, which leads into my research. And I'm going to be pretty brief on all this, but I really got into this research because I looked at the situation with morels and I realized that there's all these brilliant papers going back 100 years, and unfortunately, findings very often are attributed to the wrong mushroom. We're kind of starting back at ground floor with this taxonomic revision. That doesn't mean all those findings are invalidated. It just means that we need to do work to figure out what species were actually being described in that work. So I wanted to aim at kind of doing similar to what Ower did, just collecting morels, using the more up-to-date techniques to actually identify them, and then working them through our system of cultivation and just kind of trying to learn more about the basic biology of these mushrooms. So I started by collecting morels from all over the Pacific Northwest. This kind of started in 2017 during spring fungi. Since then, I've been collecting specimens every year. I think after this spring, I'll be close to 150 specimens. My freezer is currently full due to not being able to access the lab space right now. When I would collect these specimens, I would obviously curate them to be deposited in the herbarium, describe them to the best of my ability, take a bit of it for DNA analysis at some point in time. Also started working with freeze drying mushrooms to work as teaching specimens, which if I have a second here at the end of the presentation, I'll show you an example of that. And through my work with the evergreen culture collection and doing DNA analysis, I was kind of able to build a basic understanding of what morel cultures should look like on a macroscopic level. And I mean, this took hours of looking at morel cultures in person and through microscopes and all this other stuff to get an idea that I could visually confirm that what was growing was morels. But one thing I did notice was there was a lot of variation. I usually would end up seeing sclerotia production at some point in time. The amount of time it took to get to that point was different for every morel. And as this picture this is a great picture because each one of these is a different species of morel. And not only did I see variation between the species, but I was seeing variation within the same species, uh, which kind of led into my next bit of work, which was doing some analysis of multiple strains of the same morel, essentially. I collected a bunch of different samples of importuna from around Olympia, from landscape settings. I cultured all of them. And over a month period, I took measurements of biological characteristics, very similar to what Ower did in his initial thesis work. And through some trial runs, I established the best protocol. And what I ended up doing was growing these plates out for a month and using imaging software to assess sclerotia cover, as well as measuring hyphal growth. And what I ended up seeing was, first of all, morels grow faster than any other mushroom I have worked with in culture. These things can cover a 90 centimeter petri dish in about 3 days. It is pretty unbelievable. It really made me pause and think whether morels actually need a whole year between a disturbance and the following year to like actually fruit, just based on how quickly the mycelium grows. But again, that's true for a lot of ascomycetes. This is really the primary ascomycete I've worked with in culture, so it's just surprising compared to the basidios. There was also pretty significant difference in the sclerotia production between strains of the same mushroom, which kind of makes sense. And that was kind of the reason I did this, was because I do think this is something we're going to need to do to screen cultures for potential in indoor and outdoor cultivation. I mean, of course, based off of everything everyone said, you want a species that produces the most sclerotia. After I would culture and analyze my morels, I would grow out sclerotia in jars like I talked about, and then we were attempting to replicate Ower's indoor cultivation process. I spent from about April of last year until April of this year attempting cultivation in this beer cooler in the basement at the Evergreen State College. I tried so many different substrate mixtures. I tried mixed inoculums of several different strains. I tried single strain inoculations. I saw some really interesting stuff. I was able to assess some really interesting biological changes in response to environmental changes. What I didn't see was primordia, and what I didn't see was sexual fruit bodies, unfortunately. What I did see was a lot of Constantinella, and I saw that Constantinella seems to really come out in high humidity environments. Again, I was working with Morchella importuna, trying to adapt Ower's protocol for Rufa brunia, which here is actually our first pans of Rufa brunia that we had tried growing out. We started this in January, and unfortunately, this was a day after heavy watering. You can see this like significant Constantinella production, and then COVID happened and we got locked out. So now all of these pans are sitting in my backyard. I just thought maybe putting them out during normal morel season might give them the chance to fruit that they didn't have in the chamber, but at this point in time, nothing in my backyard has fruited. So here's some pictures of some weird growths that I was seeing throughout the process. It wasn't uncommon to see these appearing on the margins of the metal trays I was using. Here on the right-hand side, these like hyphal whorls were described by Masfini as well as Ower and Volk as being the start of primordia formation. Unfortunately, they never developed much past this. Again, I do think that's due to the fact that we were trying to adapt protocol for Rufa brunia to importuna. Doing this in the future, I'd like to work in an environmental chamber that I can actually control all the conditions much more closely. I think the key is going to be creating spring in an enclosed environment, essentially. This here was the most interesting thing that happened in this whole process. When I did get to the point of attempting to add extra nutrients to my sclerotia to get it to grow and feed, I had these hyphal aggregations that just came out of nowhere and spread like gangbusters through everything. You can see here them coming down off the side of the pan and trying to grow downwards. I don't know if this was vegetative mycelium trying to escape the pans and find new substrate to colonize. On a like cellular level, there was really no difference between this mycelium and the mycelium that would typically just be growing in the pans. There was a bit of swelling in the hyphae, but aside from that, and then over time they just kind of senesced back. But at a point in time, pretty much every tray had a growth like this. And you can see here, even a tray that I hadn't added nutrients to started to put off growths like this. This chamber was so susceptible to condition change. It was not the best place to be trying to do that work. Our work outdoors was based on the organic farm, where morels had fruited for the past few years. And based on this, we figured it was an optimal place to try to do an outdoor cultivation, kind of adopting the methods from China. We went out, we picked a spot that was kind of well away from where any of the morels had fruited in the past. What I did was I made 18-meter squared plots. I dug up the sod layer, and I left 2 meters between each plot, uncovered the sod. In a half square meter section in the center, I dug out about 3 to 6 centimeters of soil, buried my soil sclerotia mixture. This was done at about 2,000 milliliters per square meter, or per half square meter, excuse me. And then the plots were covered in cardboard and then wood chips. Now, this isn't done in China, but because of the occurrence of importuna in garden settings, I did want to test wood chips as a substrate, essentially. This could have been what prevented morels from fruiting, I'm not entirely sure, but a third of the plots ended up receiving nutrient bags just like they do in China. A third of them received no nutrient bags, and then a third of the plots actually didn't even get morel spawn, because I just wanted to see if morels would start growing in a place that there was no morels introduced to. Again, I didn't see fruit bodies, but I saw a lot of Constantinella, even before the nutrient bags were added, and it only occurred on plots that had been inoculated with morel spawn. So none of the control plots had any kind of growths like this, and it was growing, in spots of wood chips up to probably 3 or 4 centimeters deep. And it started from the point of inoculation and spread out. I noticed that when I added the bags, it definitely kind of swelled up down around the margins of the bags. By April, every plot that had been inoculated was pretty full of Constantinella all the way out to the margin. When I removed the bags, I started to see these weird kind of gelatinous growths that I thought maybe were the beginning of primordias. I questioned whether it was even Morchella at first, but I found more that were, producing morel hyphae. I isolated it back into a petri dish. It formed sclerotia. So it was definitely Morchella, but what it was beyond that, I have no idea. Some of this stuff, a lot of this was occurring right as things were getting shut down in the lab, so I haven't had the opportunity to do the microscopy and all that other stuff that I would like to do. But it was still interesting to see this kind of growth just through introducing Morchella spawn. It goes to show you that even non-sterilized wood chips are a great substrate for this mushroom. And that's the thing, if you want to try to grow morels on your own property, obviously, I just spent over a year doing this in a scientific fashion, and I failed. So it's kind of hard for me to feel like I can give you an authoritative answer on how to get this right. But I do know there's reports of spore and tissue slurries working. It's hard for me to say this as a scientist without any substantial proof, but we did use this method on the organic farm last year. And the morels that appeared this year only appeared in the spot that we inoculated with spore and tissue slurry. So if you have bad morels, extra morels, just the stems, all that stuff, just blend it up, mix it with some water, and spread it in your garden area. And if you are going to try to garden with morels, don't ever think that you can use anything other than Importuna or Rufa brunea. If anyone ever tries to sell you something else, I mean, those are the only two species that will grow in that type of setting. I'd be doubtful that even burn morels would grow in a landscape setting, even though they grow in the field sites in China. If you don't have a garden to work in, you can always just, take assessment of the trees that are around you. If you kind of are lucky enough to live in a little forested area, I mean, you can just find out what morels would grow around you well and just, identify that species and create slurries and spawn to spread in your area doing that. I think perpetuating natural morels is definitely something we could do. And this here was just kind of the leftovers of what do you do with 90 fully colonized petri dishes of morel spawn, essentially? So I just blended it all up, mixed it and diluted it out between probably 50 to 100 gallons and spread it over this entire garden space that was covered in cardboard. So this is expected to fruit next spring, and maybe I'll have the chance to report back. So just kind of wrap things up and kind of going a little over on my time, but I just want to stress the fact that there's still a lot of questions to be answered about morels. The advances we have made in the field of researching these mushrooms is quite astounding, especially in the past 5 years, and I think it's only going to progress further as time goes on. And I think it's really important to, appreciate morels not just as food. And this was something that took me so long to start doing, but you might be interacting with a morel that no one has ever seen before. And just because there's a ton of them, it doesn't mean you got to take everything. Here's a picture of a little slug I saw eating the Importuna that I decided to leave. I think we don't appreciate how much other animals enjoy mushrooms sometimes, and we get a little narrow-sighted, especially with morels, because they're so exciting to pick. But if it's bad, just leave it. If you don't need it, just leave it. If you're in a burn, maybe, watch what you're stepping on and just make sure that you're leaving things to still attract things to that area.